So thank you so much for the introduction and for inviting me to be here. I'm absolutely delighted to be talking about something which I know very little about, but which I thought was a great opportunity for me to learn more, both by doing some more research in this area and also by interacting with all of you, many of whom I'm sure have a lot more experience in this field. So traditionally, my group spends much of its time developing statistical and computational methodology for analyzing genetic association studies, but increasingly we're moving towards microbiome analysis. And so much of this talk is going to be about how we can translate some of the tools that we've developed within the context of GWAS and next generation sequencing studies to microbiome analysis. So with that being said, as many of you know, all right, approximately, we're only about 10% human. So microbes cover virtually every surface of the human body, inside and out, and they outnumber us about 10 to 1. And so if you say there's about 10 to the 13 human or self cells inside of each of us, then there's about 10 to the 14th microbes inside of us or on us as well. If you're talking about genetic material, then there's actually about 100 times more microbial genomic material than self material. And so consequently, there's tremendous interest in understanding the role of microbiome within the context of human health and diseases. And so recent technologies now facilitate the capture and measurement of the human microbiomes. And so there's two primary technologies which are commonly used. This includes sequencing of the 16S rRNA gene, which is ubiquitous among microbial taxa, but with variable regions such that you can differentiate many different species over here. And now also increasingly people are interested in doing whole genome shotgun sequencing over here. But for the most part, I'm going to focus on 16S analysis and on talking about analysis of relative abundance. So interest in so there's a lot of different outcomes that are thought to be related to microbiome, and there's been a whole plethora of papers published recently. So everything from looking at obesity and the gut microbiome to looking at response to anti-PD-1 therapy and checkpoint inhibitors, all of this is thought to be closely related and integrally tied to the microbiome. And so this has spurred a wide range of different studies across many different disease areas, across a whole host of different human conditions, and also with regard to environmental exposures, both broadly defined and as chemical and toxic pollutants and toxicants as well. A recurring theme within the context of many of these studies is to identify associations or correlations between microbial taxa in a very broad sense and these outcomes of interest. So here I'm using the term outcome very generically, and so we can talk about disease outcomes, we can talk about time to event outcomes, so censored survival times, or we can talk about even exposures, so exposure to antibiotics, probiotics, or some other variable of interest over here. And so the talk of my title is slightly off. I said that we would be addressing challenges, but really the main challenge that I really want to talk about is basically correlative analysis. And how do we correlate and associate the microbiome, in some sense or another, with these outcomes of interest? And so within this context, there's a number of different modes of analyses that can take place. And some of the common things that are done are to do individual taxon analysis, where you basically look at each individual taxon, each individual species, genus, whatever you want to look at, and correlate the abundance of that particular taxon with your outcome or phenotype of interest. We can generalize this to doing multitaxa analysis, where instead of looking at individual taxa, you now group them based on some prior biological knowledge, either functionality or perhaps using phylogeny, and associate groups of taxa with the outcome. And then another mode of analysis is to do community-level analysis, where now you're looking for entire global shifts and whether entire communities differ or vary with regard to an extrinsic variable of interest. And there's also many more sophisticated modeling approaches and strategies that can also be taken here. And so my focus is really going to be on looking at association analysis and on the problem of correlative analysis in this case. I'm largely ignoring many issues of upstream analysis, so things like processing that are very, very important, that do have implications for downstream analyses, but which are slightly beyond the scope of what I will be discussing today. So let's first look at individual taxon analysis. And so here I'm just going to talk a little bit about some of the things that I've observed and some of the things that I think we should be careful about and some of the things where I think I'd love to be able to deal with, but which I don't really know how. So individual taxon analysis, as I've said, here we're interested in associating individual taxa with outcomes of interest. And so there's two primary strategies that are taken here. So your data that come from here are basically counts. And so you can actually treat your data as counts and analyze on that scale. The alternative is that you can analyze this as relative abundance, which is basically saying you're talking about the relative abundance of individual taxa with regard to the total microbial burden over there. And for the most part, I will again focus on looking at the issue of relative abundance, but the concepts and many of these things that I will discuss generalize as well to count-based analysis. It's just too complicated to talk about too many different things here. So just very briefly, for analysis of counts, what you're doing is you're using counts for the abundance of each taxon. There's a number of problems here. The characteristics of the data are that, one, it's sparse. It's highly over-dispersed and very non-normal. There tend to be outliers. And there are other processing issues which still seem to be at least somewhat unresolved within the field, or at least controversial. I have my opinions, and I'm happy to discuss those. And then once you have these count-based data, then you can just apply, or what is commonly done is to apply a standard univariable test, like a Wilcoxon test, or you can use more sophisticated parametric approaches, such as methods developed for RNA-seq analysis. These are all commonly used and in many cases work out, though one does oftentimes need to be a little bit cautious about parametric assumptions within the context of using RNA-seq methods for count-based data. They don't always work out entirely well, and sometimes type 1 error can be somewhat inflated there. So, but that's all I'm going to say about counts. What I'm going to talk about is analysis of relative abundance. So one of the difficulties that we face in these sequencing experiments is that it's really hard to control the total number of reads. Consequently, one strategy is, instead of analyzing just on the actual counts, the actual abundances, the actual number of reads, you transform the data into proportions over here. And so then it becomes, instead of absolute numbers, some sort of percent or relative concentration of different taxa. All right, so this is an example, just a toy example. Suppose these are a bunch of different taxa that you have over here. And so this is what you would get out of there, just counts. You can just transform it into relative abundance just by dividing by the total sum, or you can use some sorts of alternative scaling approaches. But you're basically making it a relative count now. It's no longer absolute, just relative to some total or something, some internal standard going on over there. Once you have this, it's very typical to just run standard univariate tests on each of these relative abundances. So what are the challenges that we face in doing this? Well, first of all, there's the issue of dimensionality. There's a lot of taxa in many of these studies. It depends on which microbiome and which site you're studying, but oftentimes, you can end up with hundreds, thousands of different taxa that you've profiled. Sometimes it's much less than that, but oftentimes you do end up with a good number. And this is very difficult to analyze, especially since our sample sizes are oftentimes limited. So dimensionality is an issue. The data tend to be highly, highly non-normal. So what we have over here is a histogram of the relative abundance for one taxon across a number of individuals from a real data set. And then you can see that for the vast majority of people, it's that for this guy over here, it's very, very highly skewed. It's very, very non-normal. And that's something that will need to be addressed in any downstream analyses. All right. The other problem that we oftentimes face is sparsity. So what I mean by this is that you end up with a lot of zeros in your data. You have a lot of taxa which are only detected in perhaps one or two individuals in your entire study. So this histogram over here has the number of samples in which a particular taxon is detected along the x-axis, and then here is just the frequency over here. So you can see that for the vast majority of taxa, they're only detected in a very small number of individuals within your study. So for everybody else, you're going to end up with a whole bunch of zeros in your data. And so this poses a tremendous challenge for the vast majority of statistical and computational tools. We don't like data in which there's very, very low variability. Variability is information. Lack of variability means you don't have enough information to really run systematic or robust analyses. Okay. And finally, there's the inherent compositional nature of the data itself. So just to illustrate what I mean by that, let's just take a toy example over here. And so suppose you have 3 taxa, A, B, and C, all right? And what comes out of your sequencer, basically, suppose you have 100 reads coming from taxon A, the count for B is 100, and C is also 100. Then once you transform it into relative abundance, then it's about 33% for each of these guys. Now suppose you have another sample in which the count for taxon A is 200, all right? The counts for B and C have not changed and are still 100. Well, once you transform it into relative abundance, then it ends up that taxon A now has a relative abundance of 50%, and B and C, on the other hand, drop to 25%. So even though nothing has, the amount of the total number of reads of C and B may not have changed, just because A has changed, it's inducing a change in the other guys over here. So conversely, suppose everybody goes up by an equal amount, all right, then it still ends up that the relative abundance is going to be 33%, 33%, and 33%. So even though there has been a change, you're not going to be able to detect it simply because of the nature of using relative abundances. And so this is going to pose a tremendous challenge, largely from the perspective of interpretation. And so with all of these problems, sometimes we look at this and my friend said, it's just a total disaster. Microbiome data, I don't know why we're doing it. There's not much that you can do. It's just horrifying. And so my perspective on it is a little bit different. I don't think it's on the order of perhaps the Earth exploding, Armageddon, or the Great Flood, but maybe it's on the order of maybe one of these bozos getting elected into office over here. So there's, of course, also other issues, but let's talk a little bit about these things that people seem to stress out a lot over. And so in discussing these, I want to I like to think of the world as really not that bad overall. And so there's 3 things that I really want to think about in approaching these issues. The first concept is the concept of interpretability, which is anytime I run an analysis, what am I going to be able to get out of it? What sort of results will I be able to get out of it, and how will I be able to interpret these? The second thing that I'm concerned about as a statistician is validity of any association analysis. And so with regard to validity, what I'm talking about is protecting type 1 error rate, which is my false positive rate. I want to control the false positive rate at a certain level. And once I've talked about those 2 issues, then I talk about the issue of power, which is whether or not I'm going to miss true associations. And so with these 3 things in mind, let's take a look at some of these issues that have been raised. So the first one's dimensionality and sparsity. So these are certainly real problems that we face. These are certainly real challenges. But I think, at least from my perspective, they're primarily issues of power. So that's not to say they don't matter, but they're issues of power rather than what we consider statistical validity. The validity of a lot of your approaches are still, your analysis approaches are still going to be valid. You simply may be underpowered because you have way too many zeros, you don't have enough variability in your data, or because, well, your multiple comparisons burden is going to be excessive. So it's an issue of power, real, but it's usually not the first thing that I worry about. What about this issue of non-normality? Well, I say that this is potentially a serious problem. So heavy tails and influential points, we always know that these are a problem in most of the analyses that we run, even extrinsic to microbiome data. Anytime you have heavy tails, anytime you have outliers, things that don't behave well, you're always concerned about that. But that being said, normality is not a strict requirement for a lot of the statistical methods that are out there, and we've dealt with non-normal data for decades. We can apply fancy transformations, we can use non-parametric tests, though there are inherent assumptions underlying each of these ideas, but this is something that we can get around. So is it a problem? I think it is, but at the same time, I think this is something that we can get around. All right, so it's a problem, but it could affect the validity of our analyses in the sense that you can inflate type 1 error if you're using an inappropriate test, an inappropriate strategy, but it's not the end of the world. What about the compositionality? This is where I struggle seriously. I think this is definitely a problem, but that being said, I don't think it's a quote-unquote statistical problem. And what I mean by that is, as a statistician, there's really not much I can do about it. It's a problem, all right? But my analyses are going to be valid in the sense that type 1 error rate is preserved. The difficulty, however, is in what analysis you've actually done. So suppose you look at relative abundances, you apply a t-test, and you say, well, the relative abundance of this taxon is higher in people with disease than people without disease. So yes, that's the p-value from that is valid, which is to say, if I took another sample, I'm likely to see a similar association. However, the association is inherently on the relative abundance, and there's simply no way to get around that. You can't infer from there necessarily without extrinsic information. You can't go backwards and talk about absolute abundances or even concentrations that are not relative to the total abundance of your total load over there. So I say that this is a serious problem, but I don't know that there's any way that I can get around it just by using fancy statistics. It's an inherent limitation, and to do anything more, I think, is at this point asking too much from the data without bringing in additional information. And so this is just my take on some of these issues. I think that there are issues of validity, but the biggest issue is still the issue of interpretation. At the same time, however, I don't know how to get around it, and so I think it's important to just recognize it. And at the end of the day, you're going to collect data, you're going to have to do analyses, and you just make the best of how you interpret the results, and just be aware of that, all right? So the truth is, I don't think it's really that bad. I can run analyses that are valid. I can run analyses that are reproducible, that are replicable, but then one just has to be cautious about the interpretation of the results. All right. So with that being said, I do think that we can deal with validity. So then the question is, how do I improve power? So where is it that single taxon analysis is really losing a lot of power? Well, there's a number of issues here. First of all, it's the dimensionality, as I've said. So this leads to tremendous multiple testing burden. If you want to do Bonferroni and you're looking at 1,000 taxa, then suddenly you're talking about 0.05 divided by 1,000 as your significance level. And even false discovery rate can only get you so far. We have to deal a lot with sparsity. You have a lot of zeros in your data. If you have a lot of zeros and a lack of variability, it does not matter what statistical or fancy approaches you use. There simply is not enough variation. There's not enough information in your data to be able to draw significance very easily. So your power is necessarily going to be low for virtually any statistical approach out there. And this tends to be much worse when your effect sizes are modest. So perhaps your outcome is not related to particular taxa, one or two taxa, but maybe there's entire groups of taxa that are related to your outcome, each with very modest effects, individual effects. So when your effects are modest, when you have dimensionality, when you have sparsity, it's going to seriously impact your statistical power. So as an alternative strategy, one thing that has been proposed and one thing that we've been thinking a lot more about is how to do multi-taxon analysis over here. So the idea is very simple. Instead of looking at the effect of individual taxa on your outcome, what we do is we do joint analysis of the effects of multiple taxa on your outcome simultaneously. So we typically group based on phylogeny. So you might have genre within a family or species within a particular genus. You group those guys together and you test for their cumulative effect on an outcome of interest. All right, disease status or perhaps some quantitative biomarker. And there's alternative ways of doing grouping. Dave Fredericks, who we've been working with, seriously pushes grouping based on functionality, which also seems to make sense within many contexts. But the key here is instead of looking at it, you're changing the unit of analysis from an individual taxon to now a group of related taxa. And this can potentially mitigate some of the issues. Because you're no longer analyzing as many individual taxa, this reduces your multiple testing burden. Because you're now combining and aggregating over multiple taxa, which are individually rare or detected in only a few individuals, by aggregating across multiple taxa, then that effectively boosts the variation over there. And so it's reducing the effects of sparsity. And simultaneously, by aggregating across taxa, even if they have individual modest effects, by doing aggregation, you can in principle also boost their joint signal over here. Then the question becomes, how do you actually choose to do this? Well, one strategy that is currently used, whether people recognize it or not, is to do something that we call collapsing over here. So the very idea, it's very, very simple. Suppose what you do is you simply sum up the abundance of each genus within a particular family, or all of the species within a particular genus. And so instead of conducting the analysis at the genus level, you now conduct the analysis at the family level. All right? So conceptually, very, very straightforward. You're just adding up the total abundance for each of the genre over there, and then that becomes a new collapsed value. Once you have a single collapsed value, then you basically can use any single taxon analysis approach that you would have chosen to do before. So you can do things like t-test, Wilcoxon tests, all right, or linear regression. So just to illustrate, let's just suppose you have a linear regression here. So y is some sort of quantitative continuous biomarker. And you're just going to regress it on this collapsed value c and adjust for any additional covariates that you might be interested in. Epsilon here is just some random error term. So, and then once you have this, you just test whether beta 1 over here, which is the regression coefficient corresponding to the collapsed value, is equal to 0 or not. Very, very straightforward conceptually. So the features, it's easy to do, all right? It's exactly what you would be doing if you were doing a single taxon analysis at simply a higher level of taxonomy over there. It's very, very easy to interpret. If you call significance, then instead of saying, well, an individual genus is related to your outcome, you're simply saying that the overall abundance of this family is related to your outcome of interest over here. And so consequently, it's just a very, very natural generalization of what you would be doing for single taxon analysis. So very, very interpretable, very, very easy. There are some downsides, however, if you look at this more closely. First of all, in taking this sum, basically your overall sum is going to end up being dominated heavily by taxa that are more abundant, or at the very least more variable. So even though you're adding up all of the taxa in a particular group, a particular family, it could end up being that there's one or two in there that totally dominate. So even though you would like to look at the contributions from all of them, it's really only those one or two that are going to be driving the entire difference. The second issue is that it does make inherent assumptions regarding the effects of the taxa on your outcome. And we'll illustrate that in just a couple of slides. But first, here's just an illustration. So what we have here is the average composition across all individuals in a particular study for the Lacno family over here. And so there's 12 genera over here. And you can see that there's a couple over here that are much, much more abundant than the others. They're much more variable. And so if you actually create a collapsed score, then essentially the majority of what you're going to be capturing is these two guys over here. So even though you want all of them to contribute, what ends up happening is it's really the more abundant ones that contribute to this overall count. So that's just something to be aware of. The second issue here is, let's come back to our regression model. Just for illustration, you have your outcome, intercept, the effect of the family on your collapsed variable on your outcome, additional covariates and random error, and you test whether beta is equal to 0 or not to do your analysis. Well, recall your collapsed value, it's just the sum of all of the counts within a particular group. So Z here are your taxon counts for each genus. You add them up, that gives you your total family count. If we plug that back into your regression model, so we're just taking C, putting it back in there, and that gives us this equation over here. All right, Y depends on beta 0, beta times the sum of all of these individual taxon counts. All right, just expand this out, rewrite it, and what happens? Beta 0, then you end up with beta times Z1 plus beta times Z2 plus beta times Z3, all the way up to beta times ZP if there's P taxa over there. And so if you look at this model, what is the inherent assumption that is going on over here? You're basically making the assumption that all taxa are related to the outcome with the exact same effects. So mathematically, that is the inherent assumption that you're making when you do this particular form of analysis. And in practice, that may not end up being the case. You can end up with some taxa that perhaps produce a particular product, and then you can have some taxa which eat that product. So it's potentially the case that you end up with opposing directions, and those guys are actually going to cancel out in this form of analysis. And so deviations from this assumption are going to, again, seriously impact your power. So collapsing, it's valid, but in principle loses power if many of the taxa are not associated with your outcome. So recall again our assumption that all of the taxa are related to the outcome with the exact same effects. If you have some that are not, then by averaging and summing over those guys, you're going to be affecting your power. It's particularly going to lose power if you have taxa that have opposing directions of effect. What ends up when you sum up is that you just cancel out and you smear out your signal exactly. And simultaneously, you'll also lose power if it ends up being the case that the lower abundance taxa are truly driving your associations. So collapsing and just adding up, conducting the analysis at a higher taxonomic level, it makes perfect sense. It makes intuitive sense, but just be aware that there are certain situations where this form of analysis can lose some statistical power. All right. So when we first started looking at microbiome data, and this was suggested to us, we made the observation that sparsity is a problem, dimensionality is a problem, but there are key similarities between what we're doing for multitaxon analysis and the analysis of rare genetic variants in next-generation sequencing studies with germline polymorphisms. The key similarities are, first of all, we're doing multi-feature analysis, so you're testing the joint effect of multiple features at the same time, and we have to deal with extreme sparsity. You have a whole bunch of zeros that you have to deal with in this case. So the idea that we had was to very simply borrow ideas and borrow approaches that we already use for rare genetic variant analysis and apply them within the context of microbiome association analysis with multi-taxa strategies. So we propose a multivariable testing procedure using a variance components test, which I'll describe in just the next slide over here. So we can again formulate a regression model. We have our outcome y. All right, and so you could again have intercept, but then now instead of assuming that the effect of all of these taxa are the same, we give each individual taxon its own regression coefficient, its own effect over here. So you have beta 1 Z1, beta 2 Z2, and beta P ZP, where they're no longer necessarily the same. We have additional covariates or confounders that we might like to control for, and again, just a random error term. Then to test for whether this group of taxa are related to an outcome or not corresponds exactly to testing whether all of these regression coefficients are equal to 0 or not. It's just a multivariable regression. We know how to test for all of the regression coefficients simultaneously. That being said, however, the standard test requires a p degree of freedom test, which in this case may end up having a low power simply because you're wasting a lot of degrees of freedom. These guys may be correlated. And because it's also problematic because you're going to have to estimate the regression coefficients in many cases, which can be very, very difficult if you have very sparse data. If you only have one individual with a particular taxon detected, then estimating the regression coefficients can be very, very unstable over here. So as an alternative, we're going to use a strategy which is very commonly used both for genetic association analysis, gene expression analysis, and other contexts in genomics, where we basically assume that our regression coefficients come from a common distribution, or come from a distribution with mean 0, variance wj times tau. I'm sorry, there should not be a j there. Where wj is a weight that we introduce to upweight the effects of lower abundance taxa. So basically, if you have a particular taxon which is not very variable, we choose to introduce a weight that upweights it in your analysis. And then to test whether all of our regression coefficients are equal to 0 or not corresponds exactly to testing whether tau is equal to 0. So instead of doing p degree freedom test, testing p different parameters, you're now only going to test a single parameter. And so this analysis is valid because under the null, everybody's 0. So 0 is 0. 0 does come from a distribution with mean 0 and variance 0. But it can oftentimes have much better power. And so this has been used many different times in gene expression and also GWAS analysis. And this is just the particular score test that is used. So I'm not going to talk too much about all of the nitty-gritty details in this case, and I'm just going to illustrate it on a particular data set. So the data were published in Genome Biology by Morgan. It's from Curtis Huttenhauer's group. And so in this study, they collected both host genomics, in particular gene expression data, and microbiome on the same subjects. So they had 255 individuals samples. 196 microbiome samples were collected from the pre-pouch ileum, and 59 from the pouch. All right, and so on each of these samples, they measured approximately 7,000 OTUs, and we also had about 20,000 genes that were profiled in this study. To illustrate our approach, all right, and I emphasize it's really illustration in this case, we grouped genre into families, and then we randomly selected 1,000 genes just for illustration, and we associated each gene's expression with each family, and then just counted up how many that were called significant over here. And so these are the results. So at the FDR is 10% level, what we have over here is a number of genes called significant as being significantly associated with each of these families over here. So MVA is the multivariable analysis approach that we've considered, and collapsing is just the standard analysis that one might take just by adding up all of the taxon counts. And so multivariate analysis, you can see, in many cases does seem to find more things being significant. All right, so at the very least, we can show that in real data, it can call more things as significant over here, particularly for some of these families. All right, though it is not universally the case that it always wins. There are situations where it does not, but in many cases, it does seem to do a little bit better. And we have a whole host of other simulations that illustrate this. Just to show type 1 error rate was correctly controlled for each of these. Here's just an example set. We simulated 10,000 null data sets. Microbiome data were bootstrapped from the Bifidobacteriaceae family, and the outcomes are just random normals that we simulated without regard to the microbiome composition. And this is the QQ plot. This is a negative log 10 expected p-value, so it should be uniform, against what we actually observed, and it lies exactly on the line. So our type 1 error rate appears to be pretty good. We have simulations across 4 other families as well, and results are pretty much the same. So that being said, I do want to emphasize that multivariate analysis does not always win. And in particular, our proposed approach is going to lose to collapsing under the situations where more common taxa are driving the associations, and in the situation where most of the taxa are related to your outcome with the same direction or very similar effects. And so there are situations where you anticipate this could be the case. If instead of grouping based on phylogeny, you group based on function, then you think all of these taxa probably are going to be doing something very similar. And so in this situation, you think that, that this totally makes sense, that they all have similar effects. So which test then should one choose to use in practice? It's not always clear. The reason for this is that the correct test to use depends on the true state of nature. So it depends on the true relationship between these taxa and your outcome of interest. The problem with that and our inability to determine which test to use is that the truth is unknown. In fact, if you knew the truth, then you don't need to do any analysis because that's the entire point of this study. You want to try and understand what's going on. If you already knew it, there's no point in doing anything. So huge waste of energy. But given that, all right, what we said is we'd like to develop an omnibus test that seems to work well across all situations. May not always win, but it won't lose dramatically. And so the basic idea behind what we've developed is we created a weighted test. We take the statistic from doing multivariable analysis, and then we do a weighted sum with what you would get from the collapsing analysis. So QM is the score statistic for multivariable analysis. QS is a score statistic from having done collapsing. And you just take a weighted sum of these two guys. Conceptually, very, very straightforward. And then we develop an approach. So if this weight, rho, is equal to 0, then it corresponds exactly to multivariable analysis. If rho is equal to 1, it corresponds exactly to collapsing. If it's something in between, then, well, you're using some combination of these two tests at the same time. And then we can develop a very straightforward approach that searches across the range of values for rho. So you identify the best rho, and yet we can still analytically get a p-value for the association. It's a little bit technical, but I'm happy to discuss that for anybody who wants to see more alphas and betas. And so just to illustrate what happens with this omnibus test, so here we have multivariable analysis, collapsing, and the number of genes called significant, FDR 10%, using the omnibus test. And you can see from most situations, it does close to or nearly as well as the better of multivariable analysis and collapsing. So here for the bifidos, you found 2 using genes as being associated using multivariable analysis, 1 using collapsing. The omnibus finds 2. Here, collapsing found 32. And omnibus testing, it doesn't do as well in many cases as the best method, but it oftentimes does close to as well. So you sacrifice a little bit of power, but you never really lose in this case. So you're hedging against different situations. There are situations actually where it will find a few more as being called significant. The reason is that while multivariable analysis and collapsing each have situations where they do the best, the truth may actually be something in between. And consequently, by searching across a different range of combination tests, you actually do a little bit better. All right? So that's just to illustrate this strategy over here. So I'm very rapidly running out of time, but I would like to talk a little bit about community level analysis, which is a topic that we've spent a lot of efforts on very recently. So this is another mode of analysis for microbiome data, where instead of looking at the effects of individual taxa now, or even small groups of taxa, you're now making it even bigger, and you're looking to see if entire communities differ in relation to a variable of interest over here. So you're assessing the association between overall microbiome composition now and a particular outcome of interest. So the global analysis, it's focused on identifying really what we see as concerted shifts in the community profiles. And it can be more powerful if individual taxa show very modest effects, but globally they're concerted changes. So you're not going to be able to detect individual taxa or even small groups of taxa because their effects are just so modest. But by looking and aggregating across everybody, all of the taxa there, then you can see some differences over here. Some of the advantages of these types of analyses are that you can harness relationships among taxa. You can bring in ecologically informed dissimilarities. You can bring in phylogeny and anticipated relationships among the different taxa. And I should also add that this sweeps a lot of statistical unpleasantness underneath the rug. It does not mean that it's not there, but oftentimes, at least when people are doing these forms of analysis, they tend to ignore the issues more, for better or for worse. All right. So one commonly used strategy for community level analysis is what I call distance or dissimilarity-based analysis. In general, I'll use the terms distance and dissimilarity interchangeably, though there are important distinctions between the two, but they don't matter, at least for the purposes of this presentation. And so operationally, what happens is you compute an ecologically relevant distance or dissimilarity between each pair of communities, which is to say each pair of samples. So we look at my gut microbiome, your gut microbiome, and we look at how different they are using some metric, which may include some sort of phylogeny or other ecological information. And then we compare our outcomes to this dissimilarity in the communities over here. And so there's a couple of strategies for looking at whether dissimilarity in our outcomes, or how the distances between you and me in terms of our outcomes correlate with distances between you and me in our communities. One very common strategy that's used is just to visually and qualitatively assess this. So you compute the principal coordinates of the top PCs, principal coordinates analysis, and what you do is then you just plot, say, cases and controls. So in this is a real data set. The red triangles here are individuals who smoked, and the stars over here are individuals who did not. And what we're looking at here is the microbiomes from the upper respiratory tract for 60 individuals. And you can see that you do actually have pretty good separation in this case of your cases from your controls. Imperfect, but you do see quite a bit of separation, at least using a particular distance metric called weighted unifrac over here. So this strategy is very, very attractive because you can see what's going on. Oftentimes, that's the best way to convince reviewers and journals that your paper should be published. If you can see the difference, that's oftentimes the best thing. That being said, all right, if everything is just based off of qualitative statistics, then we may as well go back to the 1800s. Statistics has gone well beyond that, in part because now we can do formal inference. And so one of the limitations of this approach is that there's no formal inference being conducted here. You're just qualitatively saying, well, there looks to be a bit of separation, which in many cases is enough, but suppose that these guys had been melded together a little bit more, then it may not be as clear in some situations. Similarly, it's very difficult to visually adjust for confounders. Especially if you have things like age and gender and multiple things going into there simultaneously. So how to visually do that or qualitatively do that, not always obvious. These types of things I think are very limited when you're dealing with non-categorical variables. For continuous, you could probably dichotomize, trichotomize. But if you're dealing with things like censored survival times or longitudinal samples, then how to put all of that into here is not always clear. And one challenge that we always face is it's not always obvious which principal components you should be looking at. Maybe the first two PCs are related to gender, but then the next couple PCs would be related to your variable of interest. And if you pick the wrong ones, then you're not going to see the differences that you're really interested in. An alternative strategy, which is also commonly used, is to do some sort of distance-based permutation test, as an example, Permanova. So the idea here is you take your matrix of distances. So there's a distance between each pair of individuals, each pair of samples in your study. So it becomes an n by n distance matrix of distances. And then what you basically do is you run a MANOVA type of analysis. You partition your dissimilarity, the sources of variability there, into different sources. And then you can get an F statistic for the association between your variable of interest or variables of interest and this dissimilarity metric. And you do permutation analysis in order to assess significance and get a p-value for association. This is very commonly done, very, very useful approach. What are the challenges here? Well, first of all, there are a number of problems with covariate adjustment. Anytime you're doing permutation analysis, covariate adjustment can be highly problematic due to a number of underlying statistical assumptions which are oftentimes not satisfied. It requires some sorts of orthogonality between your covariates and your variable of interest. You have problems in choosing an appropriate distance metric. You'll also see the same thing with qualitative analyses, but which distance should you be using? There's a whole bunch of different ones that one can choose to use, and using different things, you'll end up with different results. This approach also does not accommodate more complicated outcomes. So again, censored survival times come to mind, in part because that's something that we face in some of our studies. And so how to do community level analysis with outcomes which are not simple yes or no's, case controls, or a simple continuous random variable. It's not always obvious. It's also really slow because you have to do permutation. It's not a big deal if you're only looking at one or two variables, but if you're looking at 20,000 genes, then that can be a problem, especially if you need to get tailed type p-values, or if you're doing a whole bunch of simulations for the purposes of, say, power analysis in your grant. This can be excessively burdensome in many cases. So as an alternative, what we're going to again do is bring in some tools that we've developed within the context of genetic association analysis. So we're going to use a tool called a kernel machine over here. And so it's a regression-based approach in contrast to a simple MANOVA. And so what we do in kernel regression is we regress an outcome variable on similarity between samples based on their microbiome profiles, where similarity is measured via something that we call a kernel. And so the intuition behind our approach is actually very, very similar to the intuition behind distance-based analysis. What we do is we compare similarity in microbiome between you and me to similarity in the outcome between you and me. If you and I have very, very similar microbiome profiles in some sense or another, and we also have very, very similar outcomes, and this is true for everybody in your study, then that's suggestive of an association. That's the intuition behind the approach. Similar microbiomes always correlate with similar outcomes, then that's suggestive of an association, a transitive association between microbiome and your outcome of interest. And as I'll show that this offers a number of advantages. And so we propose to use this framework for community level analysis, all right? And so this is the actual statistical model. All right, it's a non-parametric model. You have your outcome. You have some sorts of confounders that you want to adjust for. Then Z's, again, here are now your entire profile of microbiome measurements, all of your OTU counts over here. And epsilon is just a just some random error. And so the relationship between your microbiome and your outcome is specified through this function f, which Just mathematically, it lives in some functional space generated by kernel K. So without talking too much more about the details over here, you can push around a whole bunch of alphas and betas, a little bit of math, and you can actually show that this model is equivalent to a linear mixed model where your outcome depends on, again, the covariates, and then a vector of subject-specific random effects with mean zero variance variance sigma squared. And then this subject-specific random effect has mean zero, and also variance tau times k, where k is a matrix of pairwise similarities between individuals over here. And so the connection between mixed models and this sort of non-parametric kernel method facilitates testing and estimation as long as you can specify the particular kernel at hand. So and operationally, what ends up happening is you compare similarity in your outcome to similarity. You're modeling similarity in your outcome based on similarity in your microbiome profiles over here, as long as you have the particular similarity metric in mind. So then how do you develop similarities for microbiota? Very, very simple. We already have existing distances. If you just take the inverse of a distance, then it basically becomes a similarity. So dissimilarity, you just flip the sign, or you just you flip it around, and it just becomes a similarity. And so this is how you take an existing matrix of distances, and you can transform it into a matrix of similarities very, very simply over here. And so some standard distances that are used for this form of analysis and include things like the unifrac distance, where you're incorporating some aspects of the phylogeny, and you can use this by this type of transformation to get a matrix of unifrac similarities via unifrac kernel. Similar things using other types of dissimilarity measures like weighted unifrac, variance weighted unifrac, or Bray-Curtis dissimilarity. All of these can be easily transformed. And then once you have that transformation, then you can directly use the connection with mixed models to do your hypothesis testing. Nothing particularly fancy going on. But what are the features of this? Why do we want to bother transforming it into a problem of similarities and this regression setting instead of just using existing dissimilarity-based approaches? Well, it ends up that our approach, it's actually equivalent to Permanova and standard dissimilarity-based analysis in the situation where you don't have covariates and your outcome is a simple dichotomous or continuous variable. So our approach is actually just a generalization of dissimilarity-based analysis, but it offers a number of advantages. First, it allows for very, very natural covariate adjustment. You just include them in the model. Nothing very difficult going on. So you can adjust for confounders or other things that you think might be going on there. It allows for analytic p-value computation, so it's fast, all right? And it's commonly used for genetic association analysis. And this is important because that means that you bring along a lot of existing machinery. Because you can borrow this existing machinery, then we can now bring all of this existing machinery to bear within the context of microbiome analysis. So we can do things like omnibus testing, which is searching across different distances in kernels. You no longer need to choose a particular dissimilarity metric to use. We can deal with complex outcomes, such as survival times, multivariate, or even ultra-high dimensional outcomes, because people have already developed tools for doing this within the context of genetics. And we can do other types of analyses which cannot be done right now with dissimilarity analysis, such as interaction testing or other types of conditional analyses that you might be interested in. And at least that's conceptually the case. There are a number of technical adjustments that need to be made, but at least the framework is in place to do all sorts of different things. So just to illustrate some of the places where this works, just spend a couple minutes on a couple different data sets. There was one study in which I participated where we were interested in looking at fecal proteases and whether they were of microbial origin. So the outcome in this study was basically the log fecal protease levels measured in about 60 patients with IBS. And the microbiome was measured on each individual's fecal samples. And the reason why they came to me was because they were interested in adjusting for additional covariates, which they thought might be related to their, which might be related to both the protease levels as well as to the microbiota. And so they were concerned that in their distance-based and dissimilarity-based analysis, they had not done that. So the results that they brought to me were the following. So they had run unifrac analysis, got a very significant p-value. Then they'd gotten excited and tried other things and gotten less and less significant results. And so they were sort of puzzled by what approach they should use. if they wanted to be honest. And secondly, they were concerned that they didn't know how to adjust for these guys in their analysis. And so using our approach, we were able to adjust for these additional confounders, and we also were able to do an omnibus test, and these are the results that we found. We end up losing a little bit of significance in some cases, but in some cases the degree of significance actually goes up by doing covariate adjustment. And then we are also able to get an omnibus p-value that basically searches across all of these guys. Loses a little bit of power relative to Unifrac, but at the very least, you can still call significance and have done an honest analysis without too much p-hacking in this case. All right. So just another quick illustration here. We have another data set from Memorial Sloan Kettering, where basically they took stool samples from 94 patients right after they'd undergone a bone marrow transplant. And so they did 16S RNA sequencing and had counts for about 2,400 different OTUs. They were interested in doing community level analysis with where the outcomes were overall survival, so it's a censored survival outcome, and also time to severe graft versus host disease. And so so here, distance-based analysis can't be run simply because there's no approaches that it can't accommodate censoring at all. And so these were the results of using a number of different similarities. So these are the p-values looking at the association between the community level abundances and overall survival, and also grade 3 graft versus host disease. This is if you use unweighted Unifrac, a generalized Unifrac with a weighting of 0.5. It's just a particular distance dissimilarity metric, a weighted Unifrac or Bray-Curtis dissimilarity. And these are the p-values. You can see at least that that under this particular dissimilarity, you end up with quite a bit of significance, that you're quite significant, even if under the other guys you are not, at least for overall survival. Nothing pops up as interesting for grade 3 graft versus host disease. And I should emphasize that in this particular study, despite having positive results because they focus on a couple of taxa, if you had done an agnostic screen of all taxa, none of them would have been called significant after adjustment for multiple testing. All right. And so just to show what happened more with this data set, we then took the Unifrac, we then took this generalized Unifrac. We clustered the samples based off of that, and we generated 2 different profiles. And then this is just the Kaplan-Meier plot with the difference over there. That being said, I should emphasize that the p-value from this Kaplan-Meier analysis is actually, from the logarithmic test, is not statistically significant, despite appearances. And so that just further emphasizes that you can do things like clustering and doing some dimension reduction, but again, you may lose some power. So I had one more example, which is really not that interesting, and I will just end with a little bit of summary over here. So overall, microbiome analysis, it's an area of tremendous opportunity. There's been a lot of successes, but at the same time, there's a lot of analytic questions, which I think are still very, very present. Some of these, such as how to deal with the fact that your data are inherently compositional, are things which I don't know how to address, and which I think without further extrinsic information, we're never really going to be able to do much with. I think individual taxon analysis, it's very, very simple. It's almost always valid as long as you do some sorts of appropriate transformations or use appropriate methods, but it can oftentimes lose power. Multitaxon analysis is a nice alternative, but then one has to be aware that some of the standard approaches, whether or not you know you're using them, do make assumptions which don't always hold, and one can lose some power over here. The variance component strategy that we've proposed does seem to do well in a number of situations. And then we've also talked a lot about community-based analysis, and we think that using kernels offers a nice alternative to existing distance-based strategies. And with that, I just want to acknowledge some of the people who've worked on this. Shang and Anna are my current minions, and Ni Zhao just joined the faculty at Johns Hopkins. And these are some of my other collaborators, and here are some of the references. All right, thank you.
Thank you so much for this talk. I was curious about the necessity for using relative abundance. I was curious about the necessity for using relative abundance instead of absolute abundance. I think I've heard that explained before, and I can't remember if it has to do with the sampling or the technology. It seems like if you could standardize against a certain volume of a sample, you could trust the absolute abundance, and some of those relative abundance issues would go away.
In principle, yes. And so I think, but I think the reality of it is that just through the processing, you're never quite sure how many exact reads you're going to get out of there. Even if you're putting in the same volume of DNA, it's not always clear that you're going to get the same thing out. Just depends on reagents, a lot of different things.
One of the standard ways the libraries are made is based on standardising the DNA concentration. And particularly for human samples, the DNA concentration will also include the amount of human DNA that's in there. So one of the ways that we try and get around it is to quantify the amount of bacterial genomes that are in there to begin with, or at least the number of 16S copies. And then if you can standardise the number of 16S copies going into the library, you can reduce some of the compositional effects.
Yes, yes, and that's what I mean. You need to basically use some sort of extrinsic information, but if you're just starting from the raw things over there, there's no way for me to go backwards, so.
Nice talk. So you talked a lot about the issue of sparse data and a lot of zeros. So my question is that something that we can use to our advantage? Do people ever take an approach of just looking for presence or absence, just sort of binary thresholding, this bacteria is either there or not, in rare cases? Is that an analysis that you've done or other people have done? Is there any value in looking at that?
We have done that form of analysis. So basically you dichotomize and you just say, well, if you have this taxon, then we think it's terrible for you, this species, then it's terrible. If not, then it's fine. And that's certainly a very inappropriate analysis. The problem is that, again, if you use a Fisher's exact test or any statistical test where the vast majority of individuals are zero, then you simply have no power. It doesn't really matter the particular statistical approach. You can certainly conduct the analysis. I think there's inherent value to it, but your power is necessarily low. Suppose you have a situation where a particular taxon is only identified or detected in a single individual, then you end up with a contingency table with just a single cell filled, and you're never going to be able to get that.
This is on. So I think I do genetics, and so we focus mostly on what you would consider rare variant analysis, and because those are what are clinically significant. That's what we actually test for in the lab.
Right.
And so I don't know anything about microbiome, but I wonder if even if let's say you had a data set of 100 people, and let's say only one of them has one of these rare bacteria or two, well, if the outcome there is extreme, I mean, that may be telling us something very important, even if it's not statistically significant. So just I don't know anything about microbiome analysis, but that sort of might be analogous to the genetic scenario where, yes, it's very rare to have a BRCA1 or 2 mutation, but when you see it, the phenotype is very, very pronounced, and that's why we test for it clinically.
I agree with you entirely. In fact, the techniques that we're using over here are exactly borrowed from rare variant analysis. It's simply that, at least from a statistical standpoint, there's not much. But if you can qualitatively observe it, then I think that that's certainly something worth following up on.
So, and sometimes when we would need standards here, we spike standards into our sample so we know what where a standard is. And has that been tried with microbiome analysis, and how much would that solve all of the problems you're trying to solve?
So I know people spike the standards, and that does seem to be useful, but my issue is that it's not always clear to me that the amplifications always work out exactly the same. And so I don't know enough to say, other than one of my collaborators does have a lot of this data, and we sort of use it generally for QC, but we're never quite confident in it at this point. So, but that may actually be, again, the extrinsic information that we're looking for. You need to include something else that we can basically anchor our analysis to and get from there to absolute abundance.